A very warm good evening to all participants. Hope all are doing well. So here we present our scientific blog, BioPsych Biosciences, to all of you. And this is the platform where we aim to deliver many fascinating informations and basic knowledges to people all around the world. And we take some special attentions from bioscience students to conduct various events to enhance their knowledges. So we welcome you all for this webinar on antiretroviral therapy and how does it work on HIV. So with no other delay, let me introduce our speaker today, Ms. Sandhya from National Institute of Research in Tuberculosis, ICMR. She is a gold medalist student in her undergraduation, and her area of interest are in immunology, virology, genetic engineering, animal studies, molecular and cellular therapeutics. She is also the co-inventor of the patent, the entrapment of mangiferin polyphenol in polylactic-glycolic acid nanoparticles, and its in vitro anti-cancer study in lung carcinoma cell lines. So with no other delay, let's welcome Ms. Sandhya to take over the webinar. Over to Sandhya.
Hello, Akash. Am I audible?
Yeah, yes.
A minute, please. So, hello everyone. I'm Sandhya. Am I audible, Akash?
Yes, Sandhya, you're audible. I think your screen is not visible. I think it's just blank. Yeah, now it's visible.
Now it's okay. 1 minute. Am I audible now?
Yeah, you're audible. You're audible.
Okay. So, hello everyone. As Akash said, I'm Sandhya. I'm a research scholar. I'm doing my research in Department of HIV from ICMR, National Institute of Research on Tuberculosis. My lab is working towards prevention of HIV, and we are working on the eradication of HIV co-infection TB cases. So today's webinar is about antiretroviral therapy. How does that work on HIV infection? So let's move on to our webinar. So as we all know, acquired immunodeficiency syndrome is caused by human immunodeficiency virus, which is a persistent public health problem worldwide. So according to WHO and UNAIDS 2021, there were 37.6 million people living with HIV. Among them, 27.4 million people who are accessing antiretroviral therapy, and 1.5 million people are newly infected with HIV. So there are several research focusing on HIV treatment, cure, prevention, but still there is no cure for HIV. So while retroviral therapy, antiretroviral therapy found to be promising, but it does not result in cure. So which it will take long time treatment and cost high and not affordable by many. So numerous approaches have been reported, but there is no scalable solution to cure or prevent HIV as not yet found. So before moving on to our antiretroviral topic, so let's discuss some about HIV. So HIV was originated from simian immunodeficiency virus of Premex, which is a zoonotic origin. So this SIV of chimpanzees gave rise to HIV-1, and SIV of Suti mangape monkey gave rise to HIV-2 in humans. So these are the same virus, but different clade of the same virus, that is HIV-1 and HIV-2. So this HIV-1 was isolated in 1983, and HIV-2 was first isolated in 1986. they differ in epidemics, infectivity, and their genomic part also. So it's compared to HIV-1 and HIV-2, they have 55% genomic identity. Both are belongs to the same family of retrovirus in the genus of lentivirus. So HIV-1 is subcategorized
Hello.
into hello.
Excuse me, Sandhya. I'm sorry to interrupt you. Your slide is still in the first slide. Your slide is not going to the next one. You're still in the first slide. Yeah,
Uh, may I know which slide?
yeah. Now you are in the third slide, but previously you were still in the first slide.
Okay, now it's okay.
Yeah, now it's okay.
Okay. So HIV-1 is further classified into group M, N, O, P, and group M is further subclassified into A, B, C, D, E, F, G, H, K, J, K, and CRF is combined recombinant forms. But HIV-1 is the major strain which is prevalent among worldwide. HIV-2 is prevalent in West African nations, and it slowly progressed to infection. Compared to HIV-1, its infectivity is very low. That is HIV-2. So coming to structure and genome of HIV. So the mature infectious virion size is 100 to 120 nanometer. This HIV comes under enveloped virus. So this enveloped virus consists of a lipid bilayer which composing the nucleocapsid which contains two single-stranded RNA genome of size approximately 10 kb. So as you see the diagram, so the outer extracellular domain is called envelope. The envelope contains GP120 envelope protein, which is a surface unit, and GP41, which is the transmembrane unit. So this envelope is the main part, which is essential for the HIV to infect CD4 T cells. So this is the part which interact with the receptor to infect the host cell. And coming to the matrix proteins, the P17 is a matrix protein and P24 is a capsule protein. And along with the single-stranded RNA, there you have the proteins like reverse transcriptase and integrase, which are essential for the HIV to transcribe its RNA into DNA. So this is the genome of HIV-1 and HIV-2. So HIV-1 and HIV-2 both encodes structural genes like GAG, Pol, envelope, which are essential for the structural to make the structure of HIV virus. And regulatory genes like CAT and REV, they are involved in the mechanism like transcription and translation. They are act like promoter.
Hello.
Hello.
Uh, I'm really sorry, Sandhya. Like your slides are not moving. Like still you are in third slide only. I think the
Okay.
slideshow thing is not working. Maybe you cannot put the slideshow.
Okay, now it's in slide 4, I
Yeah, now
guess.
it's in four.
Are you
Yeah.
able to see? Okay. So coming to genome of HIV-1 and HIV-2. So the structural genes are GAG, Pol, envelope, which makes the structural part of the HIV gene, HIV. And then regulatory genes like CAT, REV, they are responsible for missionaries like translation, transcription, and auxiliary genes like VIF, VTR, VTU, VTX, NEF, they are helpful in the nuclear transport of viral DNA, that is reverse transcribed DNA to the nucleus for the integration. So that we will discuss in the HIV replication part. So this GAG gene encodes proteins like matrix, capsid, nucleocapsid, and P6 protein. And Pol gene encodes protease, reverse transcriptase, and integrase. And envelope protein, envelope gene encodes surface unit, as I already told. As you can see, GP120. As you can see, a GP120, this is a surface unit, and GP41, which is a transmembrane unit. So, so coming to HIV replication. So HIV replication. So we'll see these are the steps of HIV replication. This is how the HIV infects the cell. So for the HIV needs to infect the cell, it needs a receptor. So the main receptor which targeted by HIV is CD4, which is present in T cell. So CD4 T cell are the main cell which will be infected by HIV. So for the HIV to infect, it needs CD4 receptor along with the co-receptor, which is the chemokine co-receptor like CCR5 and CXCR4. Based on the morphology of the envelope, it will use either CCR5 or CXCR4. Some viruses use both the co-receptors. So first one is binding. So with the help of outer envelope GP120, it will bind to our host CD4 receptor along with the co-receptor. So for the envelope virus to fuse, so with the help of GP120 and GP41, it will fuse with our host cell. Okay, then it will release the components like single-stranded RNA, other encoded proteins to our host cytoplasm. Then the viral RNA, with the help of its the HIV's reverse transcriptase, the enzyme which will convert viral RNA into double-stranded DNA, so that it will convert into double-stranded DNA. Then with the help of accessory genes, it will transport it into the host cell nucleus. Then it will integrate it into the host cell genome. So after, with the help of an integrase, so it will get integrated into the host cell genome. With the use of host cell's machinery, that is translation, transcription, it will synthesize its mRNA for encoding proteins, and for the synthesis of progeny, it will make use of the host cell's machinery. Then it will, but from the host cell, where you can see the infectious mature virion. This infectious mature virion is now ready to infect the other CD4 T cells. So coming to HIV transmission. So HIV transmission, many people would have studied, you have studied already in your textbooks. So ways that HIV get transmitted. So 84% is due to unprotected sex. The main part where HIV will get transmitted is mucosal transmission. As you know that the morphology of genital parts, so there you have, there the HIV has to cross the mucosal barrier, then it will get into your system. Then parental transmission, this is in case of drug use and usage of contaminated needles or syringes. And perinatal transmission, it's very less, some 4%. It comes under mother to child transmission. Then occupational transmission, this transmission occurs in medical and lab settings, and very less through body fluids or blood transmission or organ transplantation cases. And it's not transmitted by mosquitoes or any other insects, or through saliva, tears, or sweat. And it will not get transmitted if you are in close contact with people who are at HIV, and through sexual activities that do not involve the body fluids, and it will not get transmitted through air. So invasion and risk of HIV. So as I already said, the major or the main transmission part is mucosal transmission. So the parts like female genital tract, male genital tract, these are the two major transmission cases of HIV. And this intestinal tract and placenta, bloodstream are the lowest cases. So here you can see how male and female gets transmitted. So 77% male gets infected from male to male sexual contact, and 12% through heterosexual contact. In case of female, 86% due to heterosexual contact, and 14% it's due to injection, drug use, or the use of contaminated needles or syringes. So coming to the transmission, so as I already said, mucosal transmission, here you can see the mechanism how it gets transmitted through mucosal barrier. So in mucosal barrier, you can see some immune cells like Langerhans cells, dendritic cells, right? So here you can see there are viral quasi-species, right? HAR5 and X4. So as I already said, some viruses capable of using R5, that is CXCR4 and CCR5 chemokine co-receptor. So during that vaginal or male genital tract transmission, it needs to cross the mucosal barrier. So once it crosses the mucosal barrier, it needs to encounter Langerhans cells, which is a subtype of dendritic cell. So it will interact with the dendritic cell. As I already said, the envelope needs to interact with the receptor, right? So it will interact with the Langerhans cells through DC sign receptor. So it will infect the dendritic cell so that it will enter into it will cross the mucosal barrier. Thereby, it will interact with the CD4 T cell. So now the CD4 T cell get infected, then it will drain into the lymph nodes where it will enter into the adaptive immune defenses state, and there it will encounter GALT, that is gut associated lymphoid tissue and lymph nodes, and there it will enter into the thoracic duct. Then it will enter into your immune response systems like spleen, brain, liver, lung, everything. Now the system will control under the HIV infection. In case of mother to child transmission, as you can see, during pregnancy time, it will cross the placenta. That is, chorionic villi is there in placenta. There also it needs to encounter dendritic cell. Then, during the labor delivery, during the delivery time, through the body secretions or the fluid secretion, it will contact with the child. Thereby, a child gets HIV from the infected mother. Then through breastfeeding, that's why the infected mother they won't suggest to feed babies. Then the third picture is for drug use. So how it will enter into your parental transmission, parental route. There you can see macrophages and dendritic cell. It will interact with the dendritic cell. Thereby, it will enter into the system to infect the CD4 T cells. So, coming to the stages and symptoms of HIV infection, there are 4 stages of HIV infection: acute primary infection, asymptomatic infection, symptomatic infection, and progression to AIDS. So, during the primary infection, that is from the exposure to HIV, so it starts from 2 to 6 weeks with the mild symptoms like fever, rash, swollen glands. There you can see in this graph. So your CD4 count will be normal. That is from some 900 to 1000 the count will be. Then your viral load will be in the lower detection level. So during that time, your infection or your viral load won't be at the detection level. So most of the people, they don't know they are get infected, even though the symptoms are there, but you don't know you are get infected or not, because the viral load at the level below the level of detection point, as you can see in the graph. Then after this primary infection, you will move on to the asymptomatic state. So in asymptomatic state, so this comes under the clinical latency, where your virus will be in the resting state. So it will hide in your immune cells like B cells, memory B cells, and T cells. It will hide in that cells, and it won't show the viral load also. There you can see the set point viral load. So it will be in the clinical latency, and your CD4 count also will gradually reduce. So in this state, it will be progressed to the AIDS. So where you can get constitutional symptoms and opportunistic diseases like cancer, co-infection with hepatitis B and TB, like these cases. If not treated, you will this will reach to the death state. So coming to HIV treatment. So HIV treatment now will be in the antiretroviral therapy, our webinar topic. So till now there is no cure for HIV AIDS. So this antiviral therapy, the goal of antiviral therapy is to suppress the viral replication and maintain the HIV viral load at the below level of detection. So there are some class of drugs which will target some major set points of HIV replication. So ART is now recommended for everyone once you get tested positive for HIV, regardless of CD4 T cell counts. So in this combined ART, there you will have 2 different class of drugs which will target different set points of HIV replication. Since if you give monotherapy, it will emerge 2 drug resistant cases. So The overall success of combined ART regimen, its ability to suppress the viral replication without the drug-resistant cases and minimize the associated costs, and it will impact the quality on life. So, highly active ART. This combined ART is interchangeably used as highly active ART. So in this highly active ART, these also use of multiple drugs on different viral targets. So this the first effective therapy. This drug you would have studied in your textbook, that is Zidovudin, which was established in 1987 by FDA. The first approved drug for HIV, which is a nucleoside reverse transcriptase inhibitor. We'll see the class of drugs now. And the main goals of ART is to they cannot eradicate the HIV infection, but as I already said, they can suppress the HIV viral load. So the ART's clinical goal is to prolong the life of the patient, and the virological load is to suppress the viral replication and reduce the viral load at the below level of detection. And coming to the immunological goal, to improve the immune response in both quantitative and qualitative, and reduction of HIV transmission. So, like there is no preventive strategies to stop HIV to transmit, but we can, by the use of ART, we can stop the transmission from one patient to one person to another. And overall, the therapeutic goal is to stop and prevent the HIV to transmit from infected person to non-infected persons. So these are the class of drugs come from the ART therapy. So nucleoside reverse transcriptase inhibitors, non-nucleoside reverse transcriptase enzyme inhibitors, nucleotide reverse transcriptase enzyme inhibitors, and protease inhibitors, integrase inhibitors, entry inhibitors. So first we'll see about nucleoside reverse transcriptase inhibitors. So nucleoside reverse transcriptase inhibitors are the main backbone of HIV treatment. These are comes under the first line drugs because of its favorable pharmacokinetic profile, and it will sustain intracellular half-life longer period, and it shows high oral bioavailability. Available in fixed dose combinations, which will be convenient to patients to adhere to the treatment. And coming to the mechanism of action, so as I said, this nucleoside reverse transcriptase inhibitor, it will target the reverse transcriptase enzyme. So this reverse transcriptase enzyme is essential for the viral RNA to convert into double-stranded DNA so that it will integrate into the host genome, right? So what it will do, the mechanism of action is it will go incorporate the what this reverse transcriptase will do. So it will use the viral RNA as a template. Then with the use of human nucleotides, it will convert the viral RNA into double-stranded DNA, right? So this drug will go incorporated in the place of human nucleotides. So there it will stop the DNA synthesis. So it acts as a chain terminator. So because of the absence of 3-OH, which is essential for the DNA elongation, so it will stop there itself. So the DNA elongation will not be there, and there won't be the conversion of double-stranded viral RNA into double-stranded DNA. So it acts as a competitive substrate inhibitor. So these are the drugs comes under NRTs. So zidovudin, lamivudin, and tenofovir, and dinarsin. So these are the drugs which comes under NRTs, and this NRTs, if it gets mutated, so over the time, if we give the same drug, that is monotherapy. So that's what they suggested, to give combined drugs which targets different sets of different set points of viral replication. So if the reverse transcriptase gets mutated, so this nucleoside inhibitors will not get incorporated in the site, so it will start the synthesis of DNA, then viral RNA will get converted into double-stranded DNA. So this non-nucleoside reverse transcriptase enzyme inhibitors, here this will target the enzyme allosteric site. See, this nucleoside reverse transcriptase inhibitors, it will go incorporate at the site of DNA synthesis. but this will target the reverse transcriptase enzyme itself. So this non-nucleoside inhibitors, it will go bind at the enzyme's allosteric site, which is a non-competitive inhibitor. So where it will go bind at the allosteric site and it will stop the activity of enzyme itself. So this, if it gets, in case of monotherapy, if it get mutated, then DNA synthesis will start, then it will convert into double-stranded DNA. So HIV-2 is naturally resistant to non-nucleoside reverse transcriptase enzyme inhibitors. So there are two generations of NNRTs because the first generation drugs are which can develop resistance quickly. So as you can see, I have given example of efavirenz. As you can see, it will bind at the allosteric site of this reverse transcriptase enzyme, where it will stop the daughter DNA strand to synthesize. So this second generation NNRTs, they are flexible, and even though it gets resisted, it will counteract effectively. So coming to third class of drug called nucleotide reverse transcriptase enzyme inhibitors. So as the name suggests, nucleotide reverse transcriptase, so it should be the analog of nucleotides, right? So this drug, tenofovir, which is an analog of adenosine 5' monophosphate, so this will go incorporate at the nucleotide instead of ATP, it will go incorporate at the ATP site. So this drug is available as tenofovir disoproxil fumarate, which is a prodrug. Once it will enter into your system, it get hydrolyzed in liver, then it will convert into active drug, then it will start doing its action. So next form of drugs is protease inhibitors. This protease enzyme is encoded by the HIV genome. This protease enzyme is most essential for proteolytic cleavage of proteins encoded by the HIV genome. So as I already showed that, see here you can see the GAG gene encodes 4 different proteins, and COL gene encodes 3 proteins, and envelope encodes 2 proteins, right? So these proteins will synthesize is, for example, they will synthesize in 160 amino acid length, and for that to proteolytic processing, this protease enzyme will be essential to cut them into the respective parts. So this protease inhibitors will target the protease enzyme, so there won't be a cleavage of the proteins encoded by the GAG, COL, and envelope. So your virus also will loses matureness, and it will not be a mature virus. So once it gets brought off from the system, it will be an immature virus which it can't infect the host cell. And development of resistant to the protease inhibitors is high. This is the mechanism, as you can see. So the protease will go in, the protease inhibitors will go bind at the protease, and it will stop the activity of protease. Then your virus will be immature. It is not enough to infect your CD4 T cell. So these are the drugs recommended under the protease inhibitors. Then integrase inhibitors. So there is an enzyme called integrase, which are helpful for your viral transcribed DNA to get integrated into your host genome, right? So this integrase inhibitors will target the integrase enzyme. So there are 2 groups of integrase inhibitors. So integrase SAM transfer inhibitors and integrase binding inhibitors. This integrase SAM transfer inhibitors, what it will do, it restrains the binding. That is, it will go bind at the pre-integration complex, that is transcribed viral DNA part, so that it will replace the strand, so that it will not get it will not get integrated into the genome. So integrase binding inhibitors, it will directly target the integrase enzyme. So as you can see, this raltegravir inhibits both HIV and HIV2 replication. In this picture, you can see, as I already said, this is strand transfer mechanism. So it targets the position where this proviral DNA gets integrated into the genome. So now this raltegravir does not allow the proviral DNA to integrate. The proviral DNA is the sense which is the transcribed viral DNA. Um, once it integrates into the genome, it's called proviral DNA. So the last class of drugs are entry inhibitors. Entry inhibitors are more, they will focus on chemokine receptor and fusion inhibitors. This chemokine receptor inhibitors, as I already mentioned, there are two kinds of chemokine receptor used by the HIV to infect the cell. So CCR5 and CXCR4. Here you can see CCR5 receptor, marovirov, and fusion inhibitor enfuvirtide. So these class of drugs, it will act at the level of entry itself. So it will stop at the infection site itself. So as you can see, this marovirov, what it will do, it will go bind at the CCR5. So virus which uses this receptor, co-receptor for its entry, what it will do, it will stop the virus to entry into infect the cell. And enfuvirtide, so this one, so this stops the fusion of the HIV. So as I said, with the help of envelope GP120 and GP41, this virus will make use of the CD4 receptor and it will start fusion with the host cell. So with the help of enfuvirtide, it will not get fused to the host cell. Other inhibitors like maturation inhibitors, these are under development. This maturation inhibitors, what it will do, so for the virus to mature, there are some proteins, they should assemble at the site of the progeny, that is the site of the budding. So this maturation inhibitors, what it will do, it will inhibit the complex of some maturation proteins which are needed to be incorporated into the progeny virus. So it makes the virus to bud off, but the virus won't be the mature virus, so it can't infect the other cells. And other compounds which will inhibit the HIV fusion. I already mentioned that for the virus to cross the mucosal barrier, it needs to encounter Langerhans, dendritic cells, or macrophages. There you can see it needs to interact with DC sign receptor. So there is an inhibitor called BCD. It will inhibit the attachment of HIV to other immune cells. And there is a CXCR4 antagonist and CCR5 antagonist, which is a chemokine receptor inhibitors, and fusion, this ARPIN formation, which will the mechanism of fusion. So there are some inhibitors which inhibits the further fusion activities also. This is the overall inhibition strategy to stop the HIV to get replicated. As you can see, the first step is to attach, that is bind and fuse with the host cell. There you can see the entry inhibitors, this marovirov, ibalizumab. So it will target this virus not to get fused with the host cell. Suppose if it enters, so after that it will it will release its content to the cytoplasm of the host cell. Then with the help of reverse transcriptase, it will convert a single-stranded RNA to double-stranded DNA, right? Then it will form the pre-integration complex. So there you can see some components, some inhibitors inhibiting the activity of reverse transcription. Then, so step 4 is integration. So by the use of accessory genes, this pre-integration complex will move on to the nucleus. So nucleus, here you can see the integrase enzyme will be the main role playing here. So here the drugs like dolutegravir. So these drugs, what it will do, it will target the integrase and make the pre-integration complex not to integrate with the host genome. Then as I already said, this maturation inhibitors which are needed at the budding site, as you can see, budding site, you will see the proteins here. So these are the proteins which are needed for the mature infectious virus. So here it will target these complex and it will make the virus not to be infectious, which is an immature virus. So these are so coming to first line and second line ART drugs. So this first line ART drugs will consist of two NRTs, that is nucleoside reverse transcriptase inhibitors. There should be a as I already said, there should be a combination of drugs, right? So zeroudin and lamivudin comes under NRTs, that is nucleoside reverse transcriptase inhibitors. this neviperin and efavirenz, these comes under protease inhibitors. So these are comes under, so there will be a combination of drugs. And second line, so people who are not susceptible to the first line, or they show resistance against these drugs, they will move on to second line treatment. This is second line regimen. These are followed by the WHO criteria, and these regimens are followed by NACO. That is National AIDS Control Organization. As you can see, as I already said, zidovudine, lamivudine, and evaprine. These are the combination of nucleoside reverse transcriptase inhibitors and protease inhibitors, and this tenofovir and lamivudine, evaprine. So there will be a combination of drugs. They are advised to take to the HIV infected patients, regardless of CD4 counts. And then comes to this first line regimen. They are followed in adults and adolescents. And this is WHO. According to WHO, they suggested to use DTG, which is dolutegravir, which is integrase inhibitor. They are suggesting for adults, adolescent, even for pregnant people. They are suggesting to use as a second line regimen. So coming to HIV prevention, so first thing we'll come to know that if you think about HIV prevention, means you will think about vaccine. So still now there is no vaccine developed. There is no vaccine to eradicate or to prevent HIV. They are still under development. While antiretroviral treatments found to be promising to improve your life expectancy and quality of life, but decades of effort have been done to design the vaccine to fight against the HIV infection. So, but there are some unique challenges faced by the researchers to design the vaccine. The first unique challenge is lack of natural immunity to HIV. So HIV, if you there is no data, no one naturally recovers from HIV infection. See, so once you get HIV, your immune cells will be under the control of HIV. So this virus is unique, and it will not be controlled by our natural immune system. So, but some individuals have a defensive mechanism against the HIV. They are called elite controllers. These elite controllers will synthesize neutralizing antibody. This neutralizing antibody, they won't prevent HIV infection, or they won't cure HIV infection, but they will suppress the viral replication. They will make the viral load to be under the control. So they are called elite neutralizers. This neutralizing antibody, now it's a research focusing topic nowadays. So people are focusing on neutralizing antibody. This neutralizing antibody, what it will do, so it will bind at the envelope part of the HIV virus. This envelope part is the main reason, so that it will get usage of receptor of host cell. So next unique challenge is variability of HIV types. So the main drawback of vaccine design is the HIV mutation. So HIV gets rapidly mutated. So if you design a vaccine for one particular type of HIV virus, it won't be effective against the virus which gets mutated. So this is one of the major drawback of vaccine development, and lack of correlation of protective immunity. So even though there are some vaccines under development, but till now there is no data like which immune response comes under HIV infection, or how much antibody titer, and how efficient the T cells defective against the HIV infection. So these are the data where we lack in the development of vaccine, and there is no animal model which will replicate the HIV infection like our human system. Till now, researchers are performing with use of SIV, which is the where the HIV got originated. So so there are some vaccines they designed with hybrids of SIV and HIV in hoping to prevent HIV infection. So there are some approaches targeted on HIV vaccine like whole inactivator, synthetic peptide, recombinant viral vector, and virus-like particles. It's called pseudovirions, where the envelope of the pseudovirions will contain the envelope of HIV, but the genome will be of any other less virulent virus. And recombinant subunit, live attenuated, we can do HIV vaccines. So these are the pivotal vaccine candidates to date they have designed. But this RV144 provided the first signal of HIV prevention, and but it did not play a role in protecting infection against HIV. And this vaccine's efficacy is also not that much effective against the system, but still people are working on it on RV144, which was designed by Thailand people. And other vaccine approaches uh, are there. They are targeting at the site of mucosal barrier because it is the major target for HIV to get transmitted. And now research focusing on, as I already said, broadly neutralizing antibody. Now scientists are working on vaccine design to elicit the immune response of broadly neutralizing antibody. They are taking the model from elite neutralizers. These elite neutralizers, as I already said, they are some unique people will synthesize broadly neutralizing antibody. Actually, so this what will happen? So this neutralizing antibody will act on a particular virus, that is the virus which comes into a system which will establish the infection. So the neutralizing antibody will be effective against that virus which enters into your system. Again, if the virus learns the mechanism to escape, so it gets mutated. Then again, the elite controllers will synthesize the other set of neutralizing antibody which will target the mutated virus. So these people will have broad clade of neutralizing antibody. So now scientists are focusing on broadly neutralizing antibody immune responses. So they are designing vaccine in order to elicit the B cells, the memory B cells, or immune cells to synthesize broadly neutralizing antibody to maintain the viral load at the below the level of detection. So this is the recently vaccine approach made by Oxford University and this IAVI people. So these are the two vaccines are currently under research. And coming to the conclusion, so how to prevent HIV? So as we all know, we already heard about transmission. So we'll make some measures from transmission, but there is a phenomena like PEP and PREP. So PEP is post-exposure prophylaxis, PREP is pre-exposure prophylaxis. This post-exposure prophylaxis is taking HIV drugs after the exposure of HIV. Example, if you contacted with body fluids, or if you get contacted with the people who are HIV infection, so that time you came to know that people get infected. So that time you have to take some drugs after the exposure of HIV. And there are some drugs like pre-exposure prophylaxis. This pre-exposure prophylaxis, before exposure, that is, this cases comes under the medical services or clinical setting people who works on HIV. So these people will take HIV drugs before the chances of getting HIV. And there are some HIV prevention programs are there conducted by NACO, WHO, and they have some goals like biomedical interventions, behavioral interventions, and structural interventions. They will focus on how to create awareness in people who don't have knowledge about HIV, and they will give counseling, and they will work towards the people. Even though they get HIV, they has to, some main people, they has to get education, and uh, they will they will create awareness, and they will do counseling to people who get infected by HIV. So, so this is all about our topic today. So, any doubts you have, so please ask me.
Guys, if you have any doubt, you can put in the chat box or you can ask directly. Anand, do you have any doubt?
No doubts.
Hello.
Hello.
Sandhya, nice
Ah,
presentation. I
thank you.
just have a few questions. Like first, that ho- how we can see the future
Hello. Hello.
vaccine program. Hello, hello. Are you getting me? Hello.
Hello. Yeah, yeah. Tell me.
Hello.
Tell me.
Uh,
Ah,
nice presentation, first of all. So, how we
thank
can
you.
see the futuristic vaccination program? So, how we can tackle, so, as you said, neutralization antibody and other things. So, how we can prevent actually, how scientifically futuristic approach for the vaccination of HIV? How you think?
So, in my point of view, we have to design a vaccine so that it will elicit the immune cells to produce the neutralizing antibody. So, for that, we have to see the mechanism uh, elicited by the elite controllers. So, we have to see the mechanism what that elite controllers are exhibiting. From that, we can develop a vaccine. so that we'll make the antigen to present to the immune cells. In that way, it will synthesize the neutralizing antibody for that virus, for HIV virus.
Okay. So, which are the target you can find that you can in futuristic target for HIV vaccination?
So main thing is in GP 120 and GP 41, that envelope part. So in that envelope there are some N glycosylation sites which are the targets of vaccines. So there are neutralizing antibody. Till now they are targeting V3 and V2 loop of envelope. So we have to see the potential sites in the envelope. Thereby it will elicit our immune cells to synthesize neutralizing antibody against the potential targets on the envelope part.
So, okay. Okay. Thank you.
Thank you. Uh,
Shrikant Reddy has a question, I think. Like, what are the ART drug side effects?
uh, So ART drug side effects is like rashes or fever and body sickness, like that. That was based on the people also, based on the health uh, health of the people also. It will vary. So how to overcome ART drug side effects? I see ART. These drugs, all the it will based on the drugs. See, all the drugs will not be efficient. All the drugs will won't have any side effects. There are all the drugs will have some side effects, but we need to see before that we are uh, taking actually. Yeah, Soundarya, that uh, uh, Soundarya, am I audible? Hello.
Yeah, you're audible, Sandhya.
So now I'm replying to Soundarya. Uh, so comorbidities like tuberculosis, there is a regimen for people who are co-infected with TB also. They can take antiretroviral drugs, but the regimen will be different from HIV cases alone.
Guys, do you have any other doubt, or shall we conclude it? Okay, so we'll conclude the thing. Thank you, Ms. Sandhya, for this wonderful webinar. It
Thank you,
was so
Akash.
wonderful and it was so interactive also.
Thank
And
you.
guys, I just request you to fill the feedback form also, which is the link is posted in the chat box. Kindly do that. And I just over the session to Ms. Pooja, the coordinator of this webinar, to give the concluding remarks.
Hello. Hello, Akash. Is I'm
Yes, yes,
audible?
yes, Pooja.
Oh,
Yes, Pooja.
thank you, Akash. So, hello everyone. Myself Pooja, and I would like to give a vote of thanks on behalf of whole BioPsych committee to our today's webinar speaker, Ms. Sandhya, a research fellow from DB Department of HIV/AIDS, ICMR Chennai. So I would like to give a special thanks to our all listener too for attending and listening the webinar so patientfully, and I hope it satisfied you all. And at last, I will not take so much time, and I request you all to please stay tuned with us. We all are planning a lot of interesting stuff like that for you, for all, for you all. And please stay tuned and follow us on Instagram, Facebook, Spotify, YouTube, and do subscribe our Wix website. And at last, I want to give a closing remark with a message which is given by a respected Dr. APJ Abdul Kalam, and the message is like that, to the youth of our country, that he said that the two most important life goals I would like every youth to have: first, to increase the amount of time that you have at your disposal, and second, what you can achieve in the time available to you. So yeah, that's all from my side. Thank you. Over to Akash.
Thank you all, guys. We'll meet you soon in the next webinar. Thank you so much.